Hi everybody, welcome to Genetics Lab. This is going to be a recorded introduction that you can watch to the lab, and I'll make these available every week. The goal is really to save some time. So because we're splitting the lab section into smaller chunks, you'll want to kind of get yourself up to speed before you get there. And so there's a lot of documentation that's available. There's going to be the video sort of tutorials about how to do what you're going to do in the lab, and then there will be these that talk a little more about the reasons and the methodology for what you're going to be doing. So this is just an introduction to kind of the concept of Genetics Lab, and it'll ramp up to talking about the activity for the first lab. Some of you are going to be watching this to prepare for being in lab in person. Some of you are going to watch this in sort of part of your learning experience outside of the lab room. So not every bit of content is applicable to everyone, but I want to kind of give this feel of just experiencing the full experience, whether you're there in person, whether you're online, sort of being part of what's going on. So one of the topics of today is model organisms. And so that's the selection of images that's shown here. A variety of different organisms across the diversity of life that really highlight the organisms that people work on to study not only genetics, but also other aspects related to human health, medicine, and all kinds of things. So I'll usually start off with an outline like this. This should mirror or mimic the outline that you see on your Canvas page for the lab each week. And just kind of get you, I think, for this presentation, if you're watching the video, maybe you could jump around to parts that you find more applicable to you, or just, kind of give you a reference for what we're going to be covering before I get started. All right, so just an introduction, and we'll talk about model organisms, as I mentioned. And then today's lab gets you familiar with fruit flies, which will be our first model organism we work with, and also some practice working on the stereo microscope. So before you get to lab each week, there'll be some expectations for what you can do on your own. Reading the associated reading, that'll get you up to speed on what you'll be doing. Really, if you can picture yourself doing the activity ahead of time, that'll certainly help save you some time as far as knowing what to do and not wasting time sort of reading and refamiliarizing yourself with what to do. There will also be some questions and some content on the reinforcement quiz that'll be available on Canvas. You want to complete that before coming to lab. It'll be due before you get to lab. And some of the questions on there get you thinking about the concepts of what we work on each week. Participation, I shouldn't have to say this, but participation obviously is very important. Each student who's in the lab is going to be responsible for getting some part of the data, doing some of the activities. So a lot of what we do is there's activities for everyone to do. And by participation, I just want to kind of reinforce that you're an active participant and you're, I don't know, excited to be there would be great. Interested to be there is next best thing. You'll be keeping a lab notebook. We'll talk about this as we go. A lot of what we do in the genetics lab is setting the foundation for what you might do, let's say if you embark on a research career. And keeping a lab notebook is just really an important part of becoming a professional researcher. You want to keep track of what you've done, why you did it, keep track of what went wrong, interesting observations you get along the way, and really a lot of great science is done just by what people keep notes about. So if you fail to write something down, you might be missing an important part that could be important later that you didn't really know. So we'll get some practice with that. There'll be some outside lab follow-up activities. Some of them you'll be required to come back into the room and check on your experiments. Sometimes there'll be a follow-up, say, evaluation of the data that you got. Lab notebook on there twice. And same as participation, also contributing to group work. There'll be some activities where you might be working with other students on something, and then just the expectation is that you will be an active and a generous participant in your group's activities. This last one, checking in with me if you need any help. This is important not only for your own benefit of obviously doing the right thing for the lab, but also it's a good way to make sure things stay safe and on the level. So if something gets messed up, It could be that I'm not aware of that and need to be made aware of that. Sometimes the preparations, something isn't right about them and need to be fixed. Sometimes you might be making a mistake that could kind of cost you later in terms of getting good data. So if you mess something up and then you want to evaluate your data later and we find out that it was messed up, that's kind of a waste of effort and could be prevented if you check in with me about things. So I just say maybe don't be embarrassed if you think you don't know what's going on. You're probably not alone. And frequently I need reminders about when people don't know what's going on. That's a good chance that I didn't explain it well enough. So that's kind of on you to let me know that I'm making sense or not. Okay, so for the lab notebook, obviously keeping track of data, observations, you could make notes about the kind of presentations that I'm doing right here. So if I convey something important or interesting to you, you can write that down in your lab notebook. Really, there's not a danger of writing too much in your lab notebook. So sort of when in doubt, write it down. It's a good habit to get into. So we'll get some practice doing lab activities. Today we'll have a chance to look at some fruit flies, and you can make notes about what you observe and sort of what went well, what's challenging to you all throughout. These are part of what you'll be turning in for a grade. So these are maybe a little more important to you to keep track and make sure that you include in your lab notebook. So for every week before we get started, your lab notebook will need to have a purpose of the lab. So I want you to consider what we're trying to get out of each lab, kind of big picture purpose, what's the genetic lesson theme, and what is the end goal, maybe, those kind of things in your purpose. And then also a lot about your previous week. So recording your data, notes about how things went, whether you had hypotheses that you evaluated or prepared to evaluate. And then also in the summary of the previous week, just a summary, brief summary paragraph, a few sentences about what all you really learned in the previous week. So what did you do? So each lab week is going to be preparing for the current week and evaluating the previous week. Those are the big themes. As far as turning this in, I do want to encourage you to be writing in your notebook as we're meeting. So I want to make it so that you can turn in the notes that you wrote. And in my lab, you do that electronically. You can take a photo of the sheets of the lab notebook. You can take pictures and submit the pictures. You can take pictures and paste them into a Word document or something like that. You can scan them if you have access to a scanner. That might be a quick way to do it. Some people like to, but you certainly don't have to retype your lab notes. And so you would take the written notes that you had and type them up in a document. You don't have to do that. Certainly that's an easy way to turn it in electronically, but requires you a little extra work. Some people like doing that because it reinforces kind of reevaluating what you wrote down as you do that. So make sure to turn these in. They are worth some points and they're due every week, so you can get in the habit of doing that. Okay, so as far as lab safety, just in general, if you're in the lab room, there is never any food or drink allowed. You can bring in closed containers, closed container with a sandwich in there, closed container with the beverage, but I don't want you to be eating or drinking while we're in the lab room. Basically, from personal experience, there's a lot of gross stuff that goes into science labs, especially biology labs. So we work with things that give you cancer if you get exposed to them. We work with yeast and fruit flies. We work with bacteria, roundworms. There's some gross stuff that goes into the room, and you never really can tell that somebody has cleaned the space in front of you. And I don't know about you, I just don't want to think about my food being next to fruit fly guts or something like that. So the lab period, it does stretch over some time. You are welcome to eat and drink during the lab period. I'll just ask that you leave the room to do that. So you can bring those in as long as the containers stay closed, but don't partake of them while we're in the room. Proper dress for lab, closed-toed shoes, clothing that covers your skin around the areas where you might be working. We don't have a whole lot of hazards. I will present to you, I'll make you aware of the hazards that we have each week. Sometimes we have chemical hazards. Generally there's sort of like open flame and sharp glass hazards occasionally. So just be aware of those. A lot of the same precautions you would take for genetics lab as you do in chemistry lab. That includes closed-toed shoes, I think probably one of the bigger ones. for the semester. As I mentioned, there will be specific guidelines or hazards to be aware of each week. Those will be spelled out in the lab notebook for that week. And then just another, I mentioned it already once before, but this idea of letting the instructor know if you're not sure about something. So particular if something is hazardous or is say a hazardous waste disposal issue or biohazard, things like that. If you don't know that that is what you should do, if you don't know, so you've got something and you need to dispose of it and you don't know how, please feel free to ask. I don't penalize people for not knowing. It's much more in the interest of safety that we all have open communication and you feel comfortable telling me that you don't know what's going on. I'd like for you to know, but there's no grade penalty that goes along with not knowing what's going on. All right, so first topic that we go into about today's lab, we're gonna introduce the first model organism today. And so we'll talk briefly about what a model organism is. And model organisms come up in a variety of contexts, but they have been very important to genetics. So what does it mean to be a model? Well, this is a model for general principles. So you can study, for example, if you study something in a fruit fly, you are learning about genetics because the genes and DNA and cells of a fruit fly are not too different from the genes and cells and DNA of a human. And so you can study these fruit fly genes as they are involved in creating phenotypes, getting passed on between parents, and those apply to other organisms. So a lot of times it comes down to humans. A lot of what we study in model organisms we're interested in applying to ourselves, but we're also learning some very broad principles for how life works. So everything that's alive has DNA. And so if you learn how DNA gets turned into proteins, then you're learning about really any organism you might be interested in. So it's sort of a model for the bigger picture. One of the reasons to study model organisms, a few reasons, I guess. If you can get a lot of scientists to study the same organism, then you're building this large body of knowledge about the same thing. So if you're studying, let's stick with the fruit fly example, somebody's going to study how eyes develop their pigmentation. Somebody's going to study how the life cycle goes and larvae develop into pupae and so on. Somebody else is going to study how a leg is formed or how hairs are formed. And all these people studying fruit flies build up this one single unified organism of understanding, as opposed to, say, the eye people study one organism and the leg people study another organism and the embryo development people study another organism. We wouldn't be able to relate these things in the same way that we can by having everybody study the same organism. Not to say that everyone in genetics studies fruit flies, but there's a large group of scientists who all study fruit flies. And there's a large group of scientists who all study roundworms and lab mice and so on. A few of the aspects that make a good model organism. Good model organisms are ones that can be used easily, cheaply. So fruit flies, a lot of good pluses there. They are small. They live in a little vial about the size of a test tube. They can be grown at room temperature. They eat, you can grow on a banana, but we're going to feed them this sort of chemical agar mixture. They have a rapid life cycle. So in 2 weeks, you go from an adult to another adult fruit fly. They produce lots of offspring, and you can do controlled genetic crosses. So you can, as we'll set up next week, you can set up a male parent of a known genotype and a female parent of a known genotype, and you can get them to cross with each other. So obviously that having that control and these other aspects of the fruit flies makes them a really good model organism. So we'll march through just a few of the high points of model organisms. Certainly this is not representative of everything that gets studied for genetics, but kind of across the tree of life, we're covering lots of different kinds of organisms. So we'll start simple with a prokaryote. Kind of the standard research bacterium is E. coli, Escherichia coli. And this kind of stands in for prokaryotes, for bacteria, so organisms with a single circular genome. There are techniques for genetically transforming. You can put genes into bacteria and study the effects there, and certainly can manipulate the effects. Very small organisms, but a good way to study kind of general genetic, cellular, molecular processes. While we're talking about bacteria, this is a virus that infects bacteria. So virus is not technically alive, but they do have genetic material, DNA or RNA, and they have what can be described as a life cycle. So their genetic material goes into the cell, they copy themselves, and burst out of the cell. So learning how viruses work, this one's called a bacteriophage, which basically says that it's a virus that affects bacteria. People learned a whole lot about how viruses transmit by studying this virus. Moving up, we're kind of moving up toward closer and closer to humans, sort of. This is a stand-in for eukaryotic cells. So this is Saccharomyces cerevisiae. This is a yeast. This is also known as bread yeast or brewer yeast. And this is the same yeast that would be used to bake bread. And by studying these, scientists have found out about how cells divide, find out about how eukaryotic chromosomes can be arranged, and certainly easy to grow, very expendable organisms. Nobody minds if the yeast gets thrown out. A little side route here into plants. This is the model plant organism called Arabidopsis thaliana, or commonly known as Arabidopsis. And scientists have studied the molecular processes that make flowers grow or determine where a leaf should be and so on. In this plant here, we're not going to work with this one this semester, but maybe someday. We will see some of this one. This is a roundworm called Caenorhabditis elegans, or C. elegans is how it's normally described. And this is a multicellular eukaryotic animal. And so we can study how eukaryotic animals develop. So this is an interesting organism. We'll study it later. But one of the features that we have for this organism is the cell fate of a single cell dividing and dividing and dividing, and then they can actually track where all of those divided cells end up in the adult organism. Pretty cool there. This is the one we'll work with today, Drosophila melanogaster, or Drosophila, or fruit fly. And this one is, again, a multicellular eukaryotic animal, and kind of stand in for human genetic transmission. They have XY sex determination that's sort of analogous to how humans have their sex determination. And as we'll see, they're pretty easy to work with, inexpensive, small. Moving up into organisms a little more closely related to us, these are vertebrates. So this is a fish called the zebrafish, Daniorario. And you can study processes that relate to vertebrate animals here. Moving up into vertebrate mammals, the standard lab mice, Mus musculus, gets used for, if you wanted to study something a little more specific to mammals, so maybe getting into drug discovery, things like that, that are a little more specific to organisms, I would say your lab mice are a little less expendable than your fruit fly, right? Nobody really minds throwing out fruit flies, but we're going to try to get a little more research value out of mice than simply having them be so expendable. And there's a whole bunch of organisms that aren't on here. If you were studying really genes or drugs, therapies that might affect humans, you can work even closer to humans and study non-human primates and so on. So again, as I said, lots of different organisms that do get studied, and basically different benefits you can get by studying each. So you want to study the organism that's the best fit for answering your question, and also maybe being most economical for that research question. Okay, so the last thing we'll do today is to explore the stereo microscope and use it to look for some differences among fruit flies. So we'll get to experience the fruit flies today. And this is similar, not exact, to the microscopes you'll have in the classroom. There will be a separate video that introduces you to our stereo microscopes. The stereo part refers to the fact that it has two ocular lenses. So you put one on each of your eyes. And good one to get practice with. You want to separate out those ocular lenses so that they fit your eyes correctly. The focus is over here. This is kind of a crude device. This focus actually just moves the microscope up and down, and then having the right distance from the object you're trying to magnify gets you in focus. You can zoom. The ones we have in the room are have a 1x or they have 1x, and they have either 2x or 4x objective lenses, and so you can change those by rotating the knob on here, and that's your zoom. So you have basically two kinds of zoom, far out and closer in. And the 1x, 2x, and 4x, of course, those are multiplied for total magnification, multiplied by the 10x of the eyepiece or the ocular. So 1x and 2x are then total magnification of 10x and 20x. So you put your item down here on the stage. The microscopes we have options to light from below or light from above, or actually a combination of both. So depending on what you're looking at, you want the light coming from the best way to view that. And a big advantage of this kind of scope, we call the dissecting microscope. Big advantage is that you can put your item on the stage and actually manipulate it while it's there. So we're going to be brushing fruit flies around. You can touch them, flip them over. Not something you would do with a compound microscope that has just the microscope slide on there. So this works at smaller magnification, but important for boosting the view of something that you might want to actually manipulate. Okay, so some of you will be in person. I just want to introduce this to you. If you are taking this remotely, you might want to just become familiar with what we'll be doing in the room. So we'll be getting familiar with the stereo microscope. There's a chance to sort of touch all the parts that move, get the adjustment right. If you get the adjustment right, this is really a valuable way to spend more time on the microscope. If you don't have your magnification, your zoom, your distance between your eyes, if you don't have those adjusted correctly, then you're going to have some trouble actually spending a lot of time using the microscope. It gets uncomfortable and you can get some eye strain from that. So if you do it correctly, it becomes a really useful tool you can keep using for quite some time. All right, so we'll get some flies that have been anesthetized. So fruit flies, you can, it's another one of their advantages. You can anesthetize them and move them around while they're not moving, and they will wake up from that. And you're gonna look at those and decide what you wanna make for differences, and sort your flies into different piles depending on what kind of phenotype you wanna sort them by. And you'll be done once you get your flies sorted and checked by the instructor. And then when you're done with your flies, we have a fly morgue, which is a flask full of ethanol, and we'll put the adult flies in there when we're done. That's just sort of a clean way to dispose of them. And of course, as always, every week we're going to clean up when we're done. So clean up your area, put away your microscope, and leave it as clean or better than you found it today. All right, see you in lab.